Hey, and welcome to the first week of Microbiology Labs. This week we are going to focus on the essentials of lab safety guidelines and lab procedures. And we are going to really be looking at what happens when you are in a real or physical lab. So when you were here and doing what we call a wet lab, because a lot of the techniques and a lot of the principles that you learn when you were in a wet lab will apply to the clinical settings or into future classes. So when we talk about lab safety, safety is going to start with proper handling of materials and equipment. So that means proper hand washing techniques, safe handling of contaminated materials, general lab conduct, and even understanding those biosafety levels. And these are all things that are going to also be covered in your Connect lab. So, why is lab safety so important? It's because of what we refer to as microbes being ubiquitous, which means that they are found everywhere. You're going to find microbes even in environments that you would consider to be non-habitable, that nothing can live. You will find some forms of microbes. Plus, wherever you look, microbes, when you find them, they're found in such large numbers. So for these reasons, we really need to understand lab safety so that we aren't spreading microbes and we aren't contaminating areas that we should not be. If we were in that wet lab or the physical lab, one of the things that I go through with students is how do you properly remove gloves? Because when you are in a lab setting, you are wearing gloves, just like if you were in clinicals or other areas. So how do you remove gloves properly so that you're not contaminating them? Because if you've been working with your gloves on, think about it. The outside of those gloves will have microbes because they came in contact with that. So notice here, what we're trying to do is when we take a glove off, we're trying to turn it inside out without touching it on our bare skin or surface. So that's the goal here. So notice here in the first picture, the gloved hand is pinching a piece of the glove and pulling it away, and then it's pulling it down, and when it pulls it down, it's going to pull it down and the glove goes inside out. But it's always glove to glove, so no bare skin touching contaminated gloves. But now you have one gloved hand and one bare hand, so how do you do that? Notice here that the fingers go on the inside of the glove where the contaminant should not have been because it's inside. It's going to flip it down. Notice the hand is holding the contaminated glove, so then when you flip it down and flip it inside out, then you have the clean side of the glove is out and inside the glove is the other contaminated glove. And therefore we're able to then keep all the contamination contained. And then what we're going to do with it is we're going to properly dispose of it by throwing it into an autoclave red container that you can find in labs or patients' rooms and things like that. So all contaminated materials have to go into autoclave containers. That way we get them properly disposed of. So that's going to include your gloves, your petri dishes, cotton swabs, etc. So like paper towels and stuff, those usually won't go in there, but if they had to clean up a spill or something, then they would. Things that are glass in nature, so slides, etc., those go into the sharps container just like any needles would. And broken glass, of course, would go into the glass containers. But if you're in a lab setting or patient's room or something, if you break something, always notify the lab director or the charge nurse or something to make sure that there's not more that you need to do than the traditional cleanup. Recognize too when you're working in lab settings that if you are on site that there has to be proper dress code so that you would be closed-toed shoes, you cover your waist, you don't wear sleeveless shirts and things like that, but you also are going to have to make sure your clothes are not so loose and that your hair is pulled up, because when you are working in a microbe lab in particular, a lot of times you're using what's known as a Bunsen burner and it's got a flame, and loose clothing or loose hair can actually get caught up in that Bunsen burner and cause a safety hazard for the student. So, and you'll also notice that a lot of times in clinical settings or dental offices, things like that, people will wear caps or have their hair pulled back as well, just to prevent that from being loose and cross-contaminating. In a lab setting too, it's always important to understand that you cannot have any kinds of foods or drinks in a lab. That includes gums and candies. They are just not allowed. So that helps prevent contamination and keeps everyone safe. So in lab settings, that is something that if you were walking into a lab on campus and you had a drink in hand, the professor's going to tell you to either throw it out, go put it back wherever in your car, step it out into maybe a safe space in the hall, etc. So as we talk about the lab setting, and if you were in a lab setting, and even though this is online, you still have to come away with some of those concepts, again, because of the fact that they do get applied to clinical settings or to other courses that you may have for higher level sciences, etc. So when you're working in a lab, there are some safety precautions that you want to ensure, especially with your lab bench and your tools that you use. So when you are looking at a lab and you're thinking about test tubes are a lot that we do with microbiology lab. And one of the things I always remind students is be very cognizant of how you're handling a test tube. Why? Because if we're using a test tube, 9 out of 10 times it's got a liquid in that test tube. So you need to be real careful that you're setting it into a test tube rack and that it's sturdy in that test tube rack. You don't lay it on the table or you don't lean it up against something, because then it has the potential of falling over and spilling. And then if there's a spill, you're going to have to clean that up, and there's proper ways. So a lot of times you're going to put paper towels that have a disinfectant on it and let it sit, but always contact again the lab director to help you get that cleaned up. The other thing about the lab is I'm always very cautious with students to keep their work area clean. So if you walk into a lab and it's a wet lab and I'm teaching it, everybody has to put book bags up, any extra clothing up. There's no cell phones, keys, or etc. are going to be on your work area, because we're about to put microbes over there. So anything that touches that workbench has the potential of becoming contaminated. So in fact, even in the lab setting, pens and pencils don't go from the desktop to book bags, etc., because of the fact that you can, if you're writing with gloved hands and you're writing in your lab papers or whatever, then guess what? You just contaminated a pen, and then you just put it in your bag. Maybe you went home and you have a small child who loves playing with mom's or dad's stuff and pulls that pen or pencil out and sticks it in their mouth, etc. So again, these things are easier to understand a lot of times when you're in lab and thinking about it, but do want to make you very aware of some of those safety precautions that you would have to do if you were on site. One of the other things to always recognize is when you are entering, especially a microbiology lab, there's certain protocols you need to put into place. These definitely can be extrapolated to like a clinical setting and going into a patient's room. So whenever you are entering the lab, you're not going to touch anything. The first thing you're going to do is wait and figure out what your instructions are for the day. So many times students will come in, they're going to put their materials up, etc., and then they're going to wait for the instructor to tell them what each and every chemical or each and every piece of equipment is, because understanding what you're walking into is one of the best things you can do to be safe about a lab. And that's a principle that you should know even walking into, as I said, into working with a patient or somebody like that. You need to understand what it is that you are going to need to be doing. When you walk into the lab after you put things down, after that, even many times before instruction happens, students are instructed that after they set things down, the first thing they do is wash their hands and glove up. And why is that? Because they are removing any contaminants they may have on their hands. Gloving then will protect them for the duration of the lab, and then that helps keep them safe. And then they walk to their workbenches, and then they spray them down and they disinfect them. Again, walking into a patient's room, walking into a lab, whatever it is, first thing you should do is, again, know what you're supposed to be doing in that setting. Wash your hands to ensure you're not bringing in contaminants, and glove up to ensure that you're not going to spread any. And then, of course, you will properly dispose of it. Petri dishes are something that get worked with quite a bit in lab. Now, these are a little different than the test tubes we talked about earlier, because when you're dealing with a Petri dish is going to have what we call agar in it. So that makes it like a solid surface. So you can take a Petri dish, which is it's two components, so it has a lid that goes on it. And if you take the bottom of the Petri dish and you turn it upside down, the agar is not going to spill everywhere. In fact, if you actually push on agar in a live lab or a wet lab, it's going to feel very much like Jell-O. So it's got some resistance to it. You push too hard, and yes, you can break the surface. But we oftentimes work with Petri dishes. You will see them in various types of clinical and lab settings. But Petri dishes, one of the things that you have to worry about is how do you handle them. So when you are working and making a petri dish to grow a culture, for instance, you'll cover your petri dish, and then you're going to use a term we refer to, or a substance called parafilm. And parafilm is very much like a saran wrap, but it's made specifically for labs. It's a lot more durable, and you cover that because if you can see here on this picture, the top portion of the petri dish, the lid, is a little bit larger than the bottom portion because it needs to have airflow for the bacteria to grow. So we put this film around it because when it does allow for growth, a lot of times there's condensation, so you can get some water and things like that. Plus you also don't want any other contaminants to get in or to get out. So parafilm is used when we work with petri dishes to help keep them safe. So a couple things, because we've hit on a few little things here and there, and just to put it all into perspective. So when you were talking about lab procedures, when you first walk into the lab, you should be storing all your personal items properly and safely. Then you're going to go in and you are going to wash your hands. Then you're going to put on gloves and eye protection if necessary. Make sure your hair is pulled back, and then you're going to disinfect your workstation. Usually we do that with about a 10% Clorox bleach solution. Then after the instructions come from the faculty member, then you're going to grab your materials for the day and you're going to begin your lab procedures. After you've completed your lab procedures, then you got to get ready to leave the lab, and there's also a process. So before leaving the lab, while you're still gloved up, you're going to do it in like the reverse order. So then you're going to wipe down your lab bench with a 10% Clorox solution again. You're going to dispose of everything appropriately in the biohazards and those autoclave containers. Put your specimens into the incubator, and if it's a petri dish, you're going to wrap it with that parafilm. And something I failed to mention, and you actually turn it upside down and you put it in that incubator. And then after everything is cleared off of your lab station, a lot of times we will wipe it down again just to be twice to be safe. Then students are to remove their gloves in the proper technique, inside out. They put their eyewear up, and we put those into a UV container so that they can be sterilized. And then they have to wash their hands, grab their personal materials, and then you get to leave. So again, just going through what would happen if you were in what we call a wet lab or a live physical lab. Another term from the lab that you really need to make sure that you understand is what we refer to as biosafety levels. So microorganisms, they vary in their risk factor. So there are 4 biosafety levels. BSL-1 is a low risk, not known to cause diseases for humans. So that can be some of your Bacillus subtilis, some strains of E. coli, etc. Biosafety level 2, that's a moderate risk associated with some milder human diseases, so Salmonella, C. diff, S. aureus, Hep B, things along those lines. Biosafety level 3 is a high risk, and these are serious or lethal type of diseases, and these you actually have to be generally certified to be handling things, so Bacillus anthracis and tuberculosis. And biosafety level 4, these are only handled in approved labs. There's a very large number of safety measures, and you definitely have to be approved and certified to work with these, because these are going to be very dangerous. A lot of times they're exotic, high risk, and life-threatening, such as like the Ebola virus. Now, in our in-person site labs, our wet labs, we only use biosafety level 1 as the majority. There are a few that biosafety levels 2 that we will use for demonstration. So if you're looking at a little bit, there is a CDC site that can help you with understanding those biosafety levels. So in order to get more about biosafety, personal safety, lab safety, there are some things that you need to do in Connect. The first thing is you should have already done the virtual lab tutorial. That is also going to be the very first thing, so if you haven't linked on it yet, it will help you get registered for the online lab section. But then you do need to work through that one because it kind of helps you to understand how the simulation works in the McGraw-Hill Connect. And then you're going to complete 2 lab safety procedures. So one is hand washing, talking to you about the proper protocols for hand washing, and then personal safety. Then when you're finished doing your Connects, come back to D2L and take your lab safety quiz. And that's all you will be doing for this week in lab. If you have any questions, let me know.